All right. Yeah. Thank you for being here. I'm really excited for the opportunity to get to talk today at PISCeS about something that interests me from a sort of fundamental research perspective, which is in making biologically and ecologically meaningful inferences from quantitative eDNA data. So I think we can pretty much all agree that eDNA has absolutely revolutionized the science of species detection. But that's a binary way of thinking about this data because the concentration of eDNA in an environment can provide potentially relevant ecological information. It can provide information on the spatial distribution of organisms, their reproductive timing, and potentially on their abundance. A lot of early studies in the literature ended up finding pretty strong correlations between metrics of organism abundance and eDNA concentrations. And I think this fueled a lot of sort of optimism for the potential of the method. Wouldn't it be great if we could go into an ecosystem, scoop a cup of water, and make cross-trophic level, cross-taxonomic estimates of biomass? Reality is a little bit more complicated than that, unfortunately, because the key consideration here is that a lot of those early studies were conducted in controlled laboratory settings. And as is typical with proof of concept studies. Now, there's nothing inherently wrong with that, but it's worthwhile to question to what extent is this generalizable to natural ecosystems. So we tried to address that in a meta-analysis in 2019 where we went in and basically looked at the average strength of these relationships in laboratory experiments and natural ecosystems. And we did indeed find that on average, eDNA exhibited a very strong correlation with metrics of organism abundance in laboratory experiments. But when you move to natural ecosystems, this tends to get quite a bit weaker, with abundance explaining only on a scale of about 56 to 60% of the variation in eDNA concentrations observed in those environments. So I think it is worthwhile to question to what degree can eDNA be used to reliably infer sort of unseen abundance in natural ecosystems. But I also think it is important to note that more than 90% or about 90% of the studies done on the eDNA abundance literature have found positive correlations between these metrics. And the strong correlations observed in laboratory experiments indicate a strong functional link between these two variables. So I think it's also sort of worthwhile to take a step back and answer that first question with a second one. How might we advance the science of eDNA to be able to infer abundance in natural ecosystems? Now, there are things that we can do on sort of the genetic processing and analysis side of things, but I think that a relatively understudied area is actually looking at sort of the ecology of these processes. And I think, so that's what I'm going to focus my talk today on. So I think there's kind of three key things that we can do to sort of advance the science. The first is improve our survey designs to address these types of questions. The second is to integrate eDNA dynamics in natural ecosystems. And the third is to improve our understanding of the ecology of eDNA production. So an important consideration, the first one I'm going to talk about today, is making sure that we're matching appropriately the scale of our abundance estimates and our eDNA estimates. Lentic systems are a great example of this because estimates of abundance typically operate at sort of the whole lake scale. We never really know where fish are individually within a lake at any given time, but we know, there's somewhere about 3,000 fish in a lake, for example. The important take-home here is that whole lake abundance estimates need to be compared to mean lake eDNA concentrations. The unit of study here is really kind of the lake or the population of organisms, and this has important implications for survey design. A lot of eDNA surveys employ sort of targeted survey designs because they're focused on detection. So people will go into systems and sort of preferentially sample habitat where rare species might be likely to occur. That makes sense in the context of the study objectives, but targeted surveys are going to bias these sort of ecosystem or population level estimates. What we're doing is fundamentally obtaining sort of a count, except instead of counting individuals, we're trying to count particles. So I think that we can sort of borrow from well-validated study designs in the field of ecology and population surveys in particular to start sampling eDNA to address these questions. And in particular, I think we need to start thinking about employing stratified, systematic, or random sampling designs to quantify these sort of population level parameters. The important consideration here is that the strata need to reflect the sort of focal species ecology. I work in lentic systems with salmonids a lot, for example, and I found that useful strata for me are sort of dividing ecosystems up into littoral, pelagic, and benthic zones. It's also worthwhile to question what level of sampling effort is actually needed to make an accurate estimate of these sort of lake-wide eDNA concentrations. And so we collaborated with the Bernatchez lab to actually address this question. They conducted an eDNA survey on 28 lakes in Quebec where they compared mean lake eDNA concentrations with brook trout fisheries data. The important part about this study, though, is that they actually employed stratified systematic sampling because they collected 20 samples at even intervals in the littoral zones of each of these lakes. We took this data and basically did a power analysis to analyze how many samples do we need to take to get a kind of ballpark estimate of the mean lake eDNA concentrations. And we ended up finding that the majority of lakes needed less than 15 samples with a median value between 12 and 13. But in statistics, this is what we refer to as a heavy tail distribution, and there was a small number of lakes that needed quite a substantially larger number of samples to get an accurate estimate. And based off of these findings, we would recommend sampling kind of a minimum of 20 to 25 times to get a good estimate of these sort of lake-wide population parameters. Now, when we move to lotic systems, things tend to get a whole lot more complicated real fast. The main reason for that is that discharge can be both temporally and spatially variable within and between streams. And discharge is a very important dilutive effect on eDNA. If we imagine two streams that have hypothetically the same number of individuals and same biomass of individuals, but different flow regimes, we would expect the stream that has a much higher discharge rate to have a lower concentration of eDNA. It's just going to get more diluted. So some great work out of Tal Levi's lab recognized that we don't necessarily want to know the concentration in these streams per se, but we're actually interested in sort of the total rate at which eDNA is moving downstream. And you can obtain that by basically flow correcting your eDNA concentrations using stream discharge data. We also need to take into consideration that in upstream inputs and rates of particle deposition in lotic systems, a very commonly employed survey design in these systems is to basically block off a stream reach, 100 meters long or something like that, estimate numerical abundance and biomass, and then compare that to eDNA concentrations observed at the downstream terminus of our stream reaches. The problem is that if you have organisms inhabiting upstream of your study area, they're going to produce eDNA that's going to drift down into your study area. So you need to quantify that and estimate how much of that is likely to deposit out by the time it hits your downstream terminus. And that's really hard to do. But some good work from Ariel Sjogren demonstrated that we might be able to start modeling this process by looking at the depositional velocity of eDNA, which is basically a parameter that quantifies a particle particle property that we can use to sort of estimate how likely a particle is to deposit out over a given length of stream. It's also important to bear in mind that the distribution of eDNA in streams and rivers is also very laterally heterogeneous as well, because it depends on the point of origin of the eDNA in the stream and local hydrological conditions. So it's important to take into consideration that variability. So you need to be collecting, multiple samples along a lateral transect when you're trying to do these types of studies. All this to say, lotic systems are complicated. So I suspect that, in the field moving forward, lentic systems might represent better systems to do these types of proof of concept eDNA abundance studies. It's always going to be easy to collect eDNA in these systems. That's the inherent virtue of the eDNA method. Anybody can go in, take a scoop of water, and filter it. But to do these eDNA abundance studies in natural ecosystems, we need to collect validating data on our target organisms. And lotic systems are just vastly overrepresented relative to lentic systems in the eDNA abundance literature. And I suspect the reason for that is that it's just easier to collect this validating data. I've done it tons of times myself. All you have to do is throw on a backpack electrofisher, do a mark recapture experiment, and in a day or two's worth of labor, you've generated a very robust estimate of numerical abundance and biomass. To generate equivalent data in a lentic system takes a week or two's worth of labor at a minimum, and it's really only feasible in small to medium-sized lakes. The problem with lotic systems is that it is very complicated to interpret the quantitative signals because you have so many physical, hydrological parameters that affect its distribution in these ecosystems. And in order to make biologically meaningful inferences, you basically need to first denoise that data. And this is an important consideration because eDNA production is fundamentally a biological process. And I think we need to start thinking more about eDNA production as a fundamentally physiological process as well. And that's because eDNA tends to be generated from two major sources. The shedding of cells from the surface of an organism, but I suspect anyways, more importantly, the excretion of metabolic waste product. Because these things are so intrinsically linked to the physiology of organisms, we hypothesized that we could extend a metabolic theory to model eDNA production rates. The metabolic theory of ecology posits that metabolic rates and key physiological rates can be expressed as a function of the mass of an organism raised to the power of a scaling coefficient b, with a value of b somewhere between 0 and 1. For excretion rate, it's thought to be typically somewhere between 0.6 and 0.7. So that's one pathway involved in eDNA production. What about shedding? Well, shedding is likely related to physiology, but it's also probably predominantly just a function of the total surface area of an organism. And a lot of research has demonstrated that this exact same framework can be used to model those relationships. Not only that, but the value of the scaling coefficient is pretty close to what you would expect for excretion. It's somewhere between 0.6 and 0.7. So we hypothesized we could extend this framework to model eDNA production. It's a mathy way of thinking about things, but in functional terms, we would predict that a single large fish is going to produce less eDNA than equivalent biomass of smaller conspecifics. So what does it mean to have a B value of 0.7? Well, to illustrate that, I want to talk about two special cases of the value of the scaling coefficient. If the value of the scaling coefficient is 1, what we're doing here is assuming that eDNA production scales linearly with biomass. And that's ultimately because any number raised to the power of 1 is equal to itself. Here, we would assume that eDNA production is directly proportional to differences in biomass. A fish that is 10 times the size of a smaller fish is going to produce 10 times the amount of eDNA. If we assume that the value of the scaling coefficient is equal to 0, what we're doing is assuming that eDNA production is invariant with biomass. And that's because any number raised to the power of 0 is ultimately equal to 1. So it doesn't matter how big a fish is, everything's going to produce the same amount of eDNA. If the value of the scaling coefficient is in between 0 and 1, what we're doing is assuming that eDNA production scales positively but non-linearly with biomass. A large fish is always going to produce more total eDNA, but how much more is going to depend on how close the value of that scaling coefficient is to 1. So if this is the process operating at the individual level, how do we scale that into our population level abundance metrics? Well, if individual eDNA production can be modeled as a function of the mass of an organism raised to the power of a scaling coefficient, it naturally follows that population-level eDNA production can be expressed as the sum of mass values in a population raised to the power of that scaling coefficient. And we term this metric allometrically scaled mass. I do want to highlight that this framework is actually flexible enough to model relationships between eDNA production and traditional metrics of abundance. It just so happens that traditional metrics of abundance, like numerical abundance and biomass, correspond to the special cases of the value of the scaling coefficient that I mentioned on the previous slide, with numerical abundance corresponding to a B value of 0 and biomass corresponding to a B value of 1. So we tested this hypothesis by conducting whole lake experiments where we quantified the abundance of brook trout in 9 lakes in the Rocky Mountains and collected standardized size structure data and employed a stratified sampling design to sort of estimate lake-wide eDNA concentrations. We ended up finding that integrating allometry did improve the correlation between eDNA and metrics of organism abundance in the same manner we would expect, or in the exact manner we would expect it to, by basically penalizing the expected eDNA production of populations inhabited by particularly large-bodied fish. So this represents kind of a novel way of thinking about things, and we're really interested in seeing how consistent these relationships are across taxonomic groups and other ecosystems. So we're interested in other folks sort of testing these methods in other systems as well. So the easiest way to get folks to do that is to make it easy for them to do so. So in collaboration with my supervisor and some folks with the USGS, Taylor Wilcox, we developed an online tool where folks can actually go and upload their own size structure data, population abundance counts, and quantitative eDNA data, and then using the sliding scale bar pictured here at the bottom, explore how changing the value of the scaling coefficient weakens or strengthens these relationships. And you can find that available on the National Genomics Center website. It's hosted there at the link pictured on the screen. So this is all well and good from a fundamental science perspective. We've developed a framework that can model relationships between quantitative eDNA data and traditional metrics of abundance and everything in between. But if we're interested in inferring numerical abundance or biomass from our quantitative eDNA data, that's not actually what we need. What we need is a framework that simultaneously relates those two values to the distribution of eDNA that we observe in natural ecosystems. And we hypothesize that allometry represents the key parameter that kind of unifies the relationship between numerical abundance, biomass, and quantitative eDNA signals. We demonstrated this by doing a bit of simple algebra. Now, I'm not going to work through that algebra today because I don't have time in the talk. But if you go through and do the math, these are basically the equations that you can derive, that you can use to more or less correct your quantitative eDNA data to reflect numerical abundance and biomass. The important thing about them, though, is that they actually share parameters. They share the value of this normalization constant, or slope value, which is essentially a slope value, I naught, and the value of this allometric scaling parameter, b. So we need to jointly maximize sort of in order to sort of maximize the predictive utility of eDNA, we need to jointly estimate sort of the values of these I naught coefficients and scaling coefficients to sort of predict numerical abundance and biomass. And so we did this using Bayesian models where we basically ran parallel regressions that shared parameters across them. And so we applied our framework to that Brook Trout data set that I mentioned previously. And we did end up finding, again, that the value of the scaling coefficient was probably somewhere, or the point estimate for it anyways, was somewhere around 0.75. And it did significantly improve the strength of these relationships, particularly relative to assuming that eDNA production scaled with numerical abundance, or that when b was 0. However, it is important to note that the credible intervals for our estimate of b did overlap with 1. So we were unable to distinguish whether or not eDNA production scaled allometrically or linearly with biomass. Nevertheless, we took this model and basically flipped it to determine whether or not we could differentiate metrics of population abundance based off of our quantitative eDNA data. Now for biomass, the answer to that was not really. And I think the reason for that is that we didn't have a huge gradient of biomass across our study populations. They only varied by a factor of 4. And the reason for that is that we basically used populations of brook trout that solely inhabited the lake. They were the only species there. So they're more or less at their kind of maximum carrying capacity. And from a species distribution perspective, they're basically all high biomass populations. However, when we look at how that biomass is distributed across individuals, that varied by more than an order of magnitude. And here we actually can start to differentiate between our high abundance and low abundance populations, with a sort of moderate abundance group starting to emerge. This is a model based off of only 9 lakes as well. I think if we had more data, we could start to shrink down those credible intervals further as well. So stay tuned for that, because we've got some projects ongoing that have increased sample size. So that's an example of how taking into consideration size structure is important from an interest-specific perspective. But size structure variation is also ubiquitous across species and natural ecosystems. And we know that allometric scaling affects physiological processes across species. The metabolic theory of ecology is, at its fundamental core, an interspecific theory of ecology. So we hypothesize that eDNA production will exhibit both intra- and interspecific allometry. Now, a lot of major goals of eDNA analyses is actually to infer community biodiversity. This is typically done using sort of species presence-absence counting metrics, currently. But a lot of biodiversity indices actually take advantage of relative abundance data in their estimates of biodiversity. The Shannon Index is a classic example of that. So we wondered whether we could use our sort of corrected eDNA data as a direct substitute for numerical abundance or biomass in the estimation of a Shannon Diversity Index value. So to test that, we applied our framework to a previously published New Jersey marine metabarcoding eDNA survey that paired eDNA data collection with traditional estimates of abundance derived from trawling. And we searched for the value of the scaling coefficient that jointly minimized the sum of the absolute differences between eDNA and trawl-derived Shannon values for both numerical abundance and biomass. And here again, the model performed pretty reasonably well. If you look at that figure, you can see that values of the scaling coefficient between about 0.7 and 0.9 really minimize the differences in these Shannon indices. Estimations. And the model actually, they were pretty close. Across both metrics of abundance, the sum of the absolute differences was only about 0.1 or 0.2 in this range. And so I think this is just a really cool example about how you can make ecosystem level inferences by thinking about how these processes operate at the level of the individual organism. We developed a hypothesis about how individual fish produce eDNA, scaled that up to make population level inferences, and then scaled that up further to make among species to ultimately make ecosystem level inferences. And this has got me really interested in applying the framework across different trophic levels, because I think that our framework sort of represents a way to unify how we interpret this quantitative data across these different trophic levels, because we predict that the value of the scaling coefficient is going to depend on the origin of the eDNA that we're capturing. For macroscopic organisms, the eDNA that we're capturing is eDNA that is shed or excreted into the environment. We're not catching Goliath groupers on our filters. So we would expect it to scale with these sort of physiological processes. So we would expect a scaling coefficient value somewhere around 0.75 or potentially close to 1. Multicellular, if we move down a trophic level to sort of multicellular organisms like plankton and other microorganisms, the eDNA that we're capturing is probably not, most of it's probably not extraorganismal eDNA, most of it's probably actually organismal eDNA. And so we would expect the DNA that we're recovering to scale with the biomass of these organisms in the ecosystem. And as a result, we would assume that, or predict anyways, that eDNA should scale with a B value somewhere close to 1. If we move further down a trophic level to single-celled organisms, here we're still capturing sort of whole body or whole cell, but we would expect eDNA to scale there with the number of gene copy repeats per individual. Now that is not an original insight. You have to give credit to the microbial folks who have figured that out for ages. But I do want to highlight that we can actually capture that in this framework by assuming that the value of the scaling coefficient is going to be 0, or that it scales with numerical abundance. Not only will the values of the scaling coefficient differ, but the value of these normalization constants and thus slope values of these relationships will also exhibit a trophic level and phylogenetic signal, or at least we would predict it would. Plants are a great example of that because they are pretty notoriously difficult to work with eDNA, but in the context of metabolic theory, that makes a ton of sense. They are metabolically slow organisms with hard external surfaces. If we look at sort of more mobile metazoans, and so as a result, we would expect plants to have high slope values. If we look at more mobile sort of metazoan taxonomic groups, we would predict that more metabolically slow organisms like echinoderms and turtles would have higher slope values for these relationships relative to more metabolically active organisms like teleost fish. If we move down to trophic levels where we're capturing organisms' whole body, we would expect to see much lower slope values because there's going to be a closer correspondence between, or ratio anyways, between the eDNA that we recover and their biomass in these systems. But we can still also even make predictions about what these slope values will look like amongst these taxonomic groups. Plankton, at these scales, basically we would expect the value of these slopes to be moderated essentially by average cell size differences. And we know that eukaryotes have much larger cells relative to prokaryotes, so we would predict that eukaryotes should have larger slope values. And this has just got me really excited for metazoan metagenomics. I know it is currently a very challenging approach for these types of inferences and questions because prokaryotes just dominate sequencing data. But preliminary data has demonstrated that some metazoan eDNA is recoverable from metagenomic samples. A study done in Antarctica, for example, demonstrated that about 0.2 to 0.5% of your reads are going to map back to metazoan genomes. The key implication here is that in order to get sufficient data to make these types of inferences, you have to do really, really deep sequencing. And we are not there yet, not really even close. But just to give some historical perspective, the cost of sequencing has decreased dramatically in the last 20 years. And there are now companies that are basically advertising that in the next year or two, they're going to try to release a next generation sequencing platform capable of doing a $100 genome. It's going to be really interesting to think about where we might be in 10 to 15 years when the next generation of sequencing platform gets released. This is a bit of an optimistic pie in the sky, shoot for the moon perspective, though. I have to be upfront about that. I think it's worthwhile to take a step back and kind of ground ourselves in the day-to-day reality of the systems we work with and talk about sort of the short to medium-term prospects for interpreting quantitative eDNA signals. If there's one thing I want you guys to take away from my talk today, is that you just fundamentally cannot slap together an eDNA biomass regression. This is the sort of difficulty of the method. It is difficult to interpret quantitative eDNA signals because a lot of variables can affect the distribution of eDNA in natural ecosystems. But it's also worthwhile to ask what level of precision is necessary to provide sort of utility for eDNA for these types of inferences as a tool in the molecular ecologist toolbox. And to illustrate why it's an important question, I want to compare eDNA to something like a counting fence. A counting fence is a very sophisticated piece of infrastructure that can give you individual data down to the last fish in a population. But it is also incredibly expensive to install, operate, and maintain. Contrast that to going out and taking 10 to 20 scoops of water in a lentic system. That is pennies in comparison to that. And so I think there is potential for eDNA as kind of a broad-scale screening tool to identify potentially problematic or vulnerable populations, or maybe populations that are, of sufficient size for harvesting. You can do rough inferences. And so I think it's also worthwhile to talk a little bit about the two kind of commonly used techniques to analyze eDNA data. Metabarcoding generates interspecific data for many species, and I think it's probably the most commonly used tool, sort of within a broad taxonomic group, but the quantitative data produced from metabarcoding doesn't correlate super well with the original template concentrations of eDNA observed in environmental samples. And there's a couple of reasons for that. The main one is that, even though metabarcoding uses universal primers that target conserved regions in the genome, even in those conserved regions, there's point mutations that affect the amplification efficiency of different taxonomic groups. And that's going to add a lot of noise into these relationships. Metabarcoding data will also only generate fundamentally compositional data. It's kind of an inherent limitation of the method. And so there are labs working on sort of denoising metabarcoding data. The Kelly lab is doing great work on sort of figuring out these issues, for example. But until those issues are sort of sorted, we're going to be limited to very, 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 emphasis on very rough inferences from metabarcoding data, and it'll only be relative abundance data that we can generate. Quantitative PCR and digital PCR, however, provide pretty accurate estimates and precise estimates of the original concentration of eDNA template in an environmental sample. And I think that there is evidence emerging that we can kind of make broad qualitative comparisons using these techniques essentially now. We're not the only ones finding this in our brook trout system. There was a study published recently, for example, that looked at walleye, and they found that, eDNA was not unreasonably reliable as determining whether or not walleye populations were above hypothetical fishable versus non-fishable thresholds. The problem with these methods is that they are ultimately limited to species-specific inferences. You can pretty much only do one species at a time. But there still might be value in these as kind of a broad-scale, cost-efficient screening tool to identify, populations that might need supplemental monitoring or something like that. I think a key question for the field moving forward is, can we move from sort of these qualitative inferences that I suspect that we're basically able to make now, to more quantitative inferences, rough quantitative inferences. It's always going to be rough. By accounting for the eDNA dynamics in natural ecosystems, the ecology of its production, and with good survey design, or appropriate survey design, I guess, to address these types of inferences. So with that, I'd like to thank you guys for attending and listening to my talk, and I'm happy to answer any questions folks might have.